Alaikum class. I hope you are all fine. I would like to welcome you all to the second phase of online teaching. And you all know that in this phase, you will be studying the fundamentals of genetics, which is a course which is offered by the Department of Biosciences. So the course code is Bio 231, and you all know that it's a 4 credit hour course in which 3 parts are for the theory and 1 part is for the lab. So let's get started. So here are the course content. The ones that we have already covered before the lockdown, they are green colored. The one that we are going to do today is underlined and it's in red color font. And the blue part is the one that we are going to cover during this online lectures. So far we have covered the study of life, a brief introduction to biology and genetics. We have also learned a little about the chemical foundations of life. We discussed different type of cells, cell structures, cellular organelles, and the way the cells divide by the process of mitosis and meiosis in detail. We have learned a little bit about the biological membranes and also about the macromolecules, including DNA, RNA, types of RNA, and we have also learned about the process of replication, transcription, and translation in detail. In today's lecture, we are going to study about Mendelian genetics. This lecture is going to be in different parts, and it will be like delivered to you during the first week and the second week, I guess. And after completing that, we will be learning about Mendel's law of segregation and dominance and law of independent assortment. We will also be studying the extension and modification of Mendel's laws in which we will be covering the concept of incomplete dominance, codominance, multiple alleles, lethal alleles, epistasis, and we're also going to discuss some of the examples related to them. We will also be learning about sex determination and sex-linked and sex-influenced inheritance. Extranuclear inheritance, which includes cytoplasmic and inheritance due to the genetic content which is present within the chloroplast and the mitochondria. Bacterial genetics, conjugation, transformation, and transduction will also be covered, inshallah, along with chromosomal mutations in their number and structure, with some of the common diseases which are caused because of that. And we will also be studying a little bit about the developmental genetics. So, our today's topic is about Mendelian genetics. First of all, let's look at the objectives of today's lecture. In this lecture, I will be describing the traits of pea plant that were studied by Mendel, and I will also be describing the scientific reason for the success of Mendel's experimental work. We will identify what are Mendelian crosses, and we will also evaluate the results of F1 and F2 generation from Mendelian crosses of pea plant. Genetics is the study of heredity or the passing of traits from parents to the offsprings. Gregor John Mendel set the framework for genetics long before chromosomes or genes had been identified, at a time when meiosis were not well understood. For his work, Mendel is often referred to as the father of modern genetics. Mendel selected a simple biological system, garden peas, and conducted methodological quantitative analysis using large sample size. Like many great artists, the work of Krieger Mendel was not appreciated until after his death. He is now called the father of genetics. But he was remembered as a gentleman who loved flowers and kept extensive records of weather and stars when he died. He was born on July 22nd, 1822, to a poor farming family who lived in a village in northern Moravia, which is now a part of Czech Republic. His family valued education but had little resources to send him to school, so he struggled to pay for his education. Let's quickly look at Gregor Mendel's biography to know why he is called the father of genetics.
Monk, teacher, and biologist, Gregor Mendel is known today as the father of modern genetics.
Gregor Mendel's experiments on pea plants were fundamental in establishing the field of modern genetics.
Born Johann Mendel on July 22, 1822, in what was Austria and is now the Czech Republic, Mendel excelled at school and became an Augustinian monk to further his education. He was given the name Gregor when he entered the monastery.
The monastery sent him to the university in Vienna, and that gave him the education in science, math, chemistry, and botany that he then brought back to use in his teaching.
Mendel accepted a teaching position at the monastery in Brno after completing his university studies. It was during this period he began his famous pea plant experiments.
Before Mendel, people thought you would equally inherit the traits of parent A and parent B in equal proportions.
Mendel experimented with pea plants and found that instead there were dominant and recessive traits in all organisms that were passed on statistically based upon the mixing of genes between the two parents.
This theory is now known as the law of segregation. Mendel also formulated the law of independent assortment.
The law of independent assortment addressed the fact that there are a large number of traits. These traits are passed on randomly and are independent of the other traits and of the traits of the two different parents.
The ability to explain hereditary diseases and to engineer plants with desired properties are all consequences of the fundamental theory founded by Mendel.
During his life, Gregor Mendel's research was disregarded by almost everyone. It wasn't until the early 1900s, after his death, that Mendel's work was appreciated and now serves as the basis for genetics and heredity.
In his later years, Mendel became the abbot of his monastery and spent less time with his scientific endeavors. Gregor Mendel died in 1884 at the age of 62.
Gregor Mendel's work laid the foundation for modern genetics, for modern gene therapy, and is the basis for some of the most exciting discoveries in medicine currently and in the near future.
Mendel entered the Augustinian St. Thomas Abbey and began his training as a priest, as you all know. He began studying heredity using mice, but his bishop did not like that process because it involved the study of animal sex. So he switched to plants. Mendel grew and studied about 29,000 garden pea plants in his monastery's garden, where he analyzed 7 characters of the garden pea plant, like the flower color, which was either purple or white, seed texture, whether it was wrinkled or round, seed color, whether it was yellow or green, stem length, long or short, or pod color, which may be yellow or green, as well as pod texture, which may be inflated or may be constructed, and the flower position as well, which could be axial or terminal. Based on the appearance or the phenotype of the 7 traits, Mendel developed genotypes for those traits. Because of the Mendel's work, the fundamental principles of heredity were revealed, which are often referred to as Mendel's law of inheritance. We now know that the gene carried on chromosomes are the basic functional units of heredity and the capability to be replicated, expressed, or mutated. Today, the postulates put forth by Mendel form the basis of classical or Mendelian genetics. Not all the genes are transmitted from parents to the offspring according to Mendelian genetics. But Mendel's experiments served as an excellent starting point for thinking about inheritance. Mendel made all of his observations and findings crossing individual plants. We can now view a human karyotype of all the chromosomes in an individual to visualize chromosomal abnormalities in offsprings even before birth. Shortly after Mendel proposed the trait were determined by what we now know as genes. Other researchers observed that different traits were often inherited together and thereby deduced that the genes were physically linked by being located on the same chromosome. Mendel's work was the beginning of many of the advances in molecular biology over the years. Now, before we actually start Mendelian genetics, just look at some of the important key terms that we will be using in this lecture, and you need to be familiar to these. First of all, we have genetics, and you all know, and I have already explained that genetics is the branch of biology that deals with the transmission and variation of inherited characters in a particular chromosome and DNA. Now, what are genes? They are the basic fundamental units of heredity. Alleles are alternative forms of genes, or we can also say that they are variant forms of a certain gene or a given gene. I'll be using the word phenotype and genotype. Phenotype is the physical appearance of an organism, whereas genotype this is actually specific to the alleles or the alleles relating to a specific gene or the genetic makeup of an individual. I'll be also using the term true breeding plants. These are actually the plant that always produce offsprings of the same phenotype when self-fertilized, and that is homozygote for the trait being followed. Then we have these terms homozygous. Homozygous of an individual, it's an organism in which both the copies of a given genes have the same allele, whereas heterozygote is an organism that has different copies of the alleles of the same gene. Then we have a hybrid, which is an offspring resulting from a crossbreeding different entities, like two different species or purebred plant strains. Then we have the term recessive and dominant. Recessive trait is the one which is masked by a dominant gene. A dominant one is the one that is able to mask the effect of another allele. Then we have the term filial, which is actually a generation or generations descending from a specific previous ones. And the parental generation is the generation of an organism that produces a hybrid. So now you know the key terms. Now let's look at the Mendel's experiment. You will be now watching a very good lecture prepared by Dr. Sadafazad Raja on Mendel's experiment.
This is the chapter 3, Mendelian Genetics, and today I'm going to start the topic. Mendel used a model experimental approach to study the patterns of inheritance. We have already gone through the brief history of Mendel's work and his important principles of inheritance, one of which was transmission genetics, that how the genes are transmitted from parents to the offsprings, which was one of the important postulate presented by Mendel's. However, let us talk about the experimental approaches he did for his further experimentations. The most important thing is for his hybridization experiments, he has selected a garden pea. This garden pea is having specific contrasting traits: seed shape, seed color, stem length, pod shape, pod color, flower position, etc. So based on these contrasting traits, he has selected this garden pea. However, there are some other characters which are also associated with this plant, and they are the actual reason for which he has actually chosen the garden pea plant for his hybridization experiments. Mendel showed remarkable insight into the methodology necessary for good experimental biology, and this pea plant was actually easy to grow and to hybridize artificially. This pea plant is self-fertilizing in nature, and crossbreeding experimentally is easy, and it produces well and grows to the maturity in a single season. This is one of the important point for him, because in a single season he was able to get all the results, and he has analyzed the result within a single season. Here I show you a cross between a true breeding pea plants. True breeding actually means a plant which is when self only produces offsprings with the same trait. For example, if this is the purple color of the flowers of this plant, when it is selfed, it will give you the next generation having only purple color of the flowers. Similarly, this white color flower plant. If it is selfed, it will give you the next generation having only white color of the flowers of the plant. This is the parental generation he has taken first. When he hybridized these true breeding plants, he obtained the F1 generation. In this generation he got the results that all hybrid progeny have violet flowers. All of the F1 generation of the plants were having violet or purple colors of the flowers. At that time, he again self the F1 generation to get the results of F2 generation, and he obtained the F2 generation showing the different results. Here, few of the plants were showing the white color of the flower of few of the plants. This means that these characters were transferring from F1 generation to F2 generation and have been expressing in F1 generation and then F2 generation. Only the traits which were recessive have expressed in F2 generation. However, the dominant traits have expressed in F1 generation. So this was a simplest method or the simplest cross he has made. The traits Mendel used for his experiments were seed shape, seed color, pod shape, pod color, flower color, position and stem height and so on. So based on his crossbreeds, he has got the F1 results in which it was shown that in F1 results when he crossbred the true breed of seed shape, he got all round, here all yellow, all full, all green, all violet, all axial and all tall. But in F2 generation he obtained different results, near to 3:1 ratio, 3 to 1 wrinkled, 3 round to 1 wrinkled, 3 yellow to 1 green, and so on. So these were the contrasting traits he analyzed for his experiments. There are some modern genetic terminologies he has used during the analysis of his experiments. So these modern terminologies are when both alleles are the same, capital T and capital T, or small t and small t. Actually, this character is for the length of the stem or the height of the plant. This represents tall plant and this represents short plant. When it is capital T or small t, the individual is homozygous, means having same types of alleles, either capital T or small t for this trait, or the individual plant is known as homozygote. When the alleles are different, capital T with the combination of small t, then this plant is actually heterozygous in its allelic combination, and the individual is known as heterozygote. Parents differ by a single trait. Let's suppose these are the two true breeding plants. Capital T is the allele for tall, small t is the allele for dwarf. Within the combination in this plant, capital T makes a homozygous tall plant like this, and small t is a homozygous dwarf plant like this. Phenotypically, they are same as their genotype. The monohybrid crosses reveals how one trait is transmitted from generation to generation. Mendel's simplest crosses involved only one pair of contrasting trait. Each such experiment is called monohybrid cross. A monohybrid cross is basically is made by crossing the true breeding individual plants from two parent strains, each having one of the two contrasting forms of the character. Like for tallness it is capital T, small t, and for the seed color, seed shape, flower position, and so on. Here you see this is one of the cross having a single character of tallness. This is the parental generation P1, represented by P1 or parental generation. This is truly breeding for the homozygous tallness. Both of the alleles are capital T and capital T. Here this is the short plant having both the alleles are small t and small t. This is the gamete formation, and this plant can only make one type of gamete having only capital T, and this plant can make only small t containing gamete. So by crossing these true breeding parental generation, he obtained F1 generation, and the combination of these two gametes was capital T and small t. Appearance of the plants or the phenotype was tall in F1 generation. Phenotype is actually the appearance of the plant, whereas the genotype, the combination of allele was different, that is heterozygous, having capital T and small t because of this combination. So the genotype was all heterozygous tall and phenotype is all tall. So this was the F1 generation or first filial generation. By selfing these F1 generation or by crossing F1 generation within the individuals of F1 generation, there was a different combination, because now this heterozygous can make two types of gametes carrying capital T or small t, and again this can make two different types of gametes, capital T or small t. So by making here the checkerboard, you can see here the combination. The combination can be like from this capital T and this capital T, here capital T and small t, here this small t and capital T, here this small t and small t, here. So the F2 generation or the second filial generation was having different results. The phenotype overall was 3 tall plants versus 1 short plant. So these 3 tall plants were actually physically tall, and this plant was short one. So the ratio was 3:1. Here is the phenotypic ratio 3:1, but the genotypic ratio is different. Although this is tall one and these two are again tall, but genotypically these two are different from this one. How? Because this is homozygous tall, having both capital T's in a genetic combination within this plant. But these two are heterozygotes or heterozygous tall plants having a combination of capital T and small t. And this is homozygous dwarf or homozygous short. So the ratio will be 1 homozygous tall, 2 homozygous tall, and 1 homozygous short. Another example is the parental generation truly breeding for the color of the flower. This generation was truly breeding for purple color, and this was truly breeding for white color of the flower. In F1 generation, they have received the combination of, for example, this was purple, capital P, small p, small p. In F1 generation, the combination was capital P and small p. So in a similar way, when this capital P and small p were crossed with a capital P and small p, the resulting F2 generation received this combination, capital P, capital P, capital P, small p, again capital P, small p, and this one must be small p, small p. So the ratio is 3 purple is to white, is to 1 white. This is homozygous purple. These two are heterozygous purple. This one is homozygous white. So these results have shown that there is a specific kind of ratios among these crosses, which are actually monohybrid crosses presenting only single character. Now make a cross, let us make a cross between the truly breeding contrasting seed color of the parental generation up to F2 generation. If we look at the color of the seed, you see the seed color. Let us see here. For the seed color, there was yellow and green, and yellow was the dominant one. Now we have to make a cross. For the parental plant, truly breeding for yellow seed color will be like this. Truly breeding for green color, which is recessive trait, will be like this. If we cross these both traits, let us make gametes along the sides of this checkerboard. Capital Y here of one parent, capital Y here for one parent. This is one of the parent, and the other parent will be having these gametes along this side. Now let us make the combination within the checkerboard. All these 4 individuals will receive one capital Y allele. Okay, and all these 4 individuals will receive small y as an allele, and now here is the combination. Now what you will see, all these 4 individuals are phenotypically having yellow seed color, right? Because yellow was the dominant. So all these individual plants were having yellow seed color, but they are heterozygous, all heterozygous, having different allelic combinations. This is actually F1 generation. F1 generation. So what about F2 generation? Let us make the checkerboard for F2 generation. We will take the similar combination from this F1 generation, which was represented by capital Y and small y. So we will make a cross here. Let us make the checkerboard. Capital Y, capital Y, with the combination of this small y gamete here and here. So what will be the combination? First of all, we have to write the yellow one. Yellow one will receive yellow allele from here and yellow allele from here. Then this yellow here and this yellow here. And now we will just complete the checkerboard with the green here, here, and again this individual will receive 2 of the green alleles. Now based on this checkerboard, what you can see is there are 3 individuals out of these 4 having yellow seed color, while there is only one which is having green seed color. So here the genotypic and phenotypic ratio is different. What is that? I am going to just write it down here. This is now F2 generation. And the phenotypical ratio is, mean the physical appearance is 3 yellow is to 1 green. And the genotypical ratio or the genotype is 1 homozygous yellow, 2 heterozygous yellow, and 1 homozygous green. So this is the specific ratio, or we can say this is the Mendelian ratio. 3:1 phenotypic ratio and 1:2:1 is the genotypic ratio. Now we will see the Mendel's first 3 postulates, and these postulates are unit factors are in pairs, and it says that genetic characters are controlled by unit factors existing in pairs. The unit factors are actually the genes, and the unit factors, for example, can be capital T and this small t, and they are existing in the pairs in an individual. This is the pair. Similarly, dominance and recessiveness is a property of these traits. It says that when 2 unlike unit factors like these, they are unlike. These 2 are like factors, whereas these 2 are unlike factors, responsible for a single character, are present in a single individual. One factor is dominant to the other, which is said to be the recessive. Here these are the unlike characters, right? One unit factor must be dominant, and the other, which is dominated by this factor, is said to be the recessive one, that is small t in this case. The third postulate is the segregation, which says during the formation of gametes during meiosis, the paired unit factors separate or segregate randomly so that each gamete receives one or the other with equal likelihood. Like here, if we draw these unit factors like this, for example, they are unlike unit factors. They are going to make alleles, gametes during meiosis with this capital T, with this small t. Here this parent is going to make this capital T, this small t gamete. So there is a random combination, right? This can unite here, this can unite here, this can unite with this small t, and this small t can again unite with this small t. So this combination is, or the separation is, random. If we look at the unit factors in detail, so unit factors are present in pairs, right? So in the monohybrid cross involving tall and dwarf stems, a specific unit factor exists for each trait. Each diploid diploid individual receives one factor from each parent because the factor occurs in pairs. Three combinations are possible: two factors for tall stems, two factors for dwarf stem, and one for each factor. So every individual possesses one of these three combinations which determines the stem height. These three combinations are capital T, capital T. The second combination is capital T, small t. The third combination is small t, small t. So what it says is, here you can see the homologous pairs of chromosomes. One of the chromosome is paternal from father and other is maternal from mother. During meiosis or replication, what happens is it gets duplicated and forms a pair of chromosomes, pair of homologous chromosomes. So the unit factors are present in pairs during the first meiotic prophase. Here you see this duplicated homologous chromosome might be coming from father and is having a capital G presenting the dominant trait, and this is the small g coming from mother, maybe represented by small g because it is recessive, or so on. These are the similar kind of pairs of chromosome that are homologous with one another. So these are the genes, they are present on the chromosomes which have their alleles, right? This small g has the allele capital G. This second slide is showing showing the dominance and recessiveness. This is one of the quality, or you can say this is one of the important factor during the inheritance of the characters. In each monohybrid cross, the trait expressed in the F1 generation is controlled by the dominant trait or the dominant unit factor. The trait not expressed is actually controlled by the recessive unit factor. Means the trait which is not expressing is actually the recessive one. The term dominant and recessive are also used to designate the traits in case all stems are said to be dominant over recessive dwarf stems. Like here in this table, you can see for flower color, the dominant is purple. For plant height, the tall is dominant. For seed color, yellow is dominant. Seed shape, round is dominant. For pod color, green is dominant. And for pod shape, this full or inflated is dominant. And for the flower position, axial is actually the dominant trait. Whenever these true breeding characters are crossed, the F1 generation will always give all purple, all tall plants, all yellow plants, all round, and so on, as I have discussed earlier. So this is the dominance and recessiveness. The third postulate is segregation. In segregation, if an individual contains a pair of like unit factors, for example, capital T or capital T, then all its gametes receive one of the same kind of unit factor. The gametes will produce only can have capital T. And if the individual is having a unit factor which are unlike factors, like here, if they are unlike capital T small t, then 50% of the gametes will be capital T and 50% of the gametes will be small t. So this is the random segregation of the gamete formation, right? So each gamete has a 50% probability of receiving either the tall or the dwarf unit factor. So here you can see, like in the previous figure, these are the homologous chromosomes, and now they are segregating during the first meiotic anaphase, and then there is a complete likelihood and a random segregation of capital G towards this pole and small g towards this pole. And when there are two gametes formed, one gamete will be having capital G and other 50% of gametes will be having small g. And similarly with this chromosome, 50% of the gametes will be having capital W and 50% of other gametes will be having small w. And if we see specifically here the combination, it could be like this G can go with this G can go with small w, this G can go with this, and this G can go with the small w. Like it can be vice versa, right? So there could be different combinations. So there was a Mendel's analytical approach, and the analytical approach led Mendel's to deduce that the unit factors exist in pairs because there were two contrasting traits for each of the characters he chose. This was the simple thing in only the garden pea. The question was why one of the two traits disappear in the F1 generation. Why? And the answer to this question is concluded after observation of the F2 generation, that the recessive trait and its unit factor, the recessive trait small t, small t, and its unit factor do not actually disappear in F1 generation, because in F1 generation they are in the combination of this one. They do not disappear. They were there in the genotype. They were there in their genotypic expression, but they were not expressing their character because there was a capital T with them. So they are merely hidden or masked, and only to reappear in 1/4 of the F2 offsprings. In F2 generation, they appear in one of the individual out of 4, as we have seen in the previous all crosses, monohybrid crosses. So this was the basic thing Mendel has done during his experiments. So Mendel deduced that the tall and dwarf alleles of F1 generation or F1 heterozygotes again segregate randomly into gametes. If fertilization is random, this ratio is predicted. So these ratios can be predicted, that is 3:1 phenotypic ratio and the genotypic ratio 1:2:1. Only these ratios can be obtained when these three postulates are positive for the analytical approach of his experiments.
So this was all about Mendel's experiment. Now, what are the main key takeaway points from this video lecture? First of all, Mendel studied 7 characteristics of the garden pea plants: flower color, seed texture, seed color, stem length, pod color, pod texture, flower position to the development of his laws of inheritance. Mendel used true breeding plants in his experiment, and these plants were self fertilized and they always produced offsprings with the same phenotype. Pea plants are easily manipulated. They can grow in one season and can be grown in large quantities. That's why Mendel used them. Based on his experiments and the garden peas, Mendel found that one phenotype was always dominant over the other phenotype, and that phenotype was said to be recessive for the same trait. Dominant traits are inherited unchanged from one generation to the next generation, while recessive traits disappeared in the first filial generation, but they reappeared in the second filial generation at a ratio of 3:1, and the ratio is for the 3 dominant to 1 recessive. In the F1 generation, Mendel found that one of the two options for each trait had disappeared, and all the offsprings were identical phenotypes. While in the F2 generation, the trait reappeared in one fourth of the offsprings, and the ratio was therefore 3:1.